Very good evening. So, yes, here we are going to start the major chapter, that is molecular basis of inheritance, where definitely you will get 3 questions, approximately 3 to 5 in your final NEET exam. That important this is. This is in connection with principles of inheritance and variation. Fine, right? What are the contents that you have to cover from this chapter? First is DNA structure and its packaging, DNA packaging. Then about RNA, types of RNA, RNA world you will learn, fine, right, and then starts the genetic search, genetic material search, excuse, where you will be exposed with the experiments like transformation, transduction experiments you will know, and then central dogma of molecular biology. Replication, transcription, translation, right? To elucidate the replication process, semi-conservative method of DNA replication, you will learn the experimental proof also, that is Michelson-Stahl's experiment, right ma? And then at last, after translation, where before that you will learn genetic code, after translation you will learn about human genome project and DNA fingerprinting. This is the whole of this chapter. Fine, right? So, let's get started with the DNA structure first. You are already done with this concept. Any add-ons, just add on the points and go through the class. First, you have to see the occurrence of DNA. Where does DNA occur? Okay, both in the prokaryotes and in the eukaryotes. Where does DNA occur? Fine, right? DNA is in 2 different forms in prokaryotes and eukaryotes. In most of the prokaryotes, DNA will exist as what ma? Double-stranded circular DNA. Double-stranded circular DNA. And in eukaryotes, you will have double-stranded linear DNA. Double-stranded linear DNA. Prokaryotes, it is double-stranded circular. Eukaryotes, it is double-stranded linear DNA. Here in prokaryotes, as the cytoplasm is the only matrix of the cell, it does not have any compartmentalization. So, nucleic acid is not present, cytoplasm is the only compartment it has, right ma? So, there of course, genetic material is not scattered all across the cytoplasm, it is situated at one place. This is the double stranded circular DNA is at one place of the cytoplasm. That place is addressed as nucleoid or genophore. Fine, right? Along with the genetic material or chromosomal DNA, that is nucleoid, there is extra chromosomal DNA also that is present in the cytoplasm, which is known as what? Plasmid DNA, which is what ma? Extra chromosomal DNA. Extra chromosomal DNA or plasmid DNA or non-genomic DNA. Till here it is clear, no? Now in eukaryotes you see, here DNA is linear and it is found in the nucleus, okay? So here you have double stranded linear DNA, fine, right? And here there is clear cut compartmentalization of organelles or cellular constituents in the cytoplasm, okay? One such compartment is what? Nucleus. Inside a nucleus only this double stranded linear DNA is present. So, now we are aware, no? Now we are aware of what types of genetic materials are present in prokaryote and eukaryotes and where is the occurrence of that particular DNA. Clear, right ma? Now we will move on to the central dogma of molecular biology and then we will start learning from the DNA. Fine, right? What is the central dogma of molecular biology? What is the central dogma of molecular biology? No, central dogma of molecular biology, which means replication. DNA will first get replicated, copied to mRNA, then ultimately proteins are synthesized. The central dogma of molecular biology is saying that the ultimate goal of DNA is to synthesize proteins. Fine, right? The ultimate goal of DNA is to synthesize proteins. This mechanism or location will vary both in prokaryotes and eukaryotes. First we will see in prokaryotes, right ma? In prokaryotes, DNA after replication, this gets transcribed to RNA by what enzyme? DNA dependent RNA polymerase. Okay, this is by DNA dependent RNA polymerase. Fine, right? And this will be translated to what? Proteins. All the events, replication, transcription, translation, occurs inside cytoplasm only, inside prokaryotes. Clear till here? Now we will look into eukaryotes. In eukaryotes, DNA replication occurs inside nucleus. DNA is transcribed to mRNA by DNA dependent RNA polymerase. This also takes place inside nucleus. Okay, and there is something called as post-transcriptional events that occurs only in the eukaryotic cell beta. Okay, that also occur inside the nucleus. What are they? Post-transcriptional changes. Post-transcriptional changes include, first is capping, that is 5' capping. Capping, 3' polyadenylation, and this is followed by splicing. Five' capping, 3' polyadenylation and splicing. These are the three post-transcriptional events. This will also occur inside where? Nucleus. Fine, right beta? So, replication, transcription, post-transcriptional events occur inside the nucleus in a eukaryotic cell. Now, the mature RNA or heteronuclear RNA that will excise from nucleus to cytoplasm with 5' cap, 3' polyadenylation and only exon sequences. They will enter into cytoplasm. So, translation occurs where? Inside the cytoplasm. Clear? Take down. Now, it is the scientist Temin and Baltimore these scientists, what did they do is, they have identified in some prokaryotic cells, in some viruses, not in a prokaryotic cell, in some viruses, okay, wherein that is reverse transcription, in some viruses where RNA is the major genetic material. Here RNA is the major genetic material. Fine. In those viruses which have RNA as the main genetic material, they have analyzed reverse transcription, because RNA will not directly synthesize proteins, because RNA is unstable. Okay. First it has to convert to some stable form, that is DNA. This is reverse transcription. Reverse transcription is by the enzyme reverse transcriptase. Reverse transcriptase. That is from RNA to DNA. Here this reverse transcriptase will be RNA dependent DNA polymerase. RNA dependent DNA polymerase. Clear? Any doubts anywhere? So you are clear with central dogma of mol bio with complete locations and sites of reactions both in prokaryotic cell and in eukaryotic cell. Now, what is DNA actually? You tell me what is DNA? I didn't ask you the full form. Can I address DNA or the nucleic acid as polymer of nucleotides? Is this a polynucleotide chain? Yes, this is a polynucleotide chain. First, let me put you one question. So, the concept of factors, transmission of factors, everything is clear, right? What inspired people or the scientists of that era to dwell into molecular biology? Whatever Mendel has proposed, okay, that was clear in the sense of factors. But then there is no molecular description on how the genes are getting transferred across the generations, right? So, that inspired the scientists to elucidate more on the transfer of genetic material or inheritance patterns in the molecular level. Fine, right? In molecular level, that inspired scientists to work on this. How is it happening in the molecular level? To understand that, first you have to analyze what? DNA, which is polynucleotide chain. Before DNA, the nucleic acid is identified as some acidic substance beta. Nucleic acid is identified as an acidic substance and it is named as nuclein by Frederick Mister. By Frederick Mister. This is identified as nuclein by Frederick Mister. Here we go. Sugar of DNA will be deoxyribose sugar, which is a pentose sugar. Pentose sugar is having how many carbons? 5 carbons, C1, C2, C3 and C4, C5. In deoxyribose sugar at second carbon there is no OH group, there is no oxygen group, only H will be present. That is why it is named as deoxyribose. How a single nucleotide is formed? One sugar molecule, first it will be binded with a nitrogenous base. What are the nitrogenous bases? That are aligned with a DNA nucleotide. We have 4 nitrogen bases here, 2 purines and 2 pyrimidines. Purines and pyrimidines. Purines will be adenine and guanine. Pyrimidines will be cytosine and thymine. Fine, right? And we know this adenine is binded with 2 hydrogen bonds to thymine. Guanine is binded with 3 hydrogen bonds to cytosine. Fine, right? So, here purines will have heterocyclic ring, fine, with 9 nitrogen molecules. C1 of the sugar is binded to, here C1 of the sugar is binded to N9 of purine. C1 of the sugar is binded to N1 of pyrimidine. Okay, and it is binded by what bond? N-glycosidic bond. N-glycosidic bond. Second point, phosphate group, right? Okay, phosphate group is binded to 5th carbon of the sugar. Remember, sugar along with nitrogen base is addressed as nucleoside. For example, if adenine is binding to the sugar, you will call it as deoxyadenosine. If guanine is binding to the sugar, it is deoxyguanosine. Cytosine binding to deoxyribose, deoxycytidine. Deoxythymidine, if thymine is binding. As such, these are the names for what? Nucleosides. Nucleotide means to 5th carbon of this nucleoside, phosphate group is binded. So now what is this, Nana? This is completely nucleoside. To the 5th carbon of this sugar, phosphate group is binded. This is by what bond? Phosphoester bond. You have to be clear on which carbon is binding and what is the type of bond for both glycosidic, phosphoester. So in a single nucleotide, how many bonds are present? 2. One is bond and one is, yeah, take down it clearly, you take down. Done? You are clear on nucleotide and bonds, right, ma? Now, so these are the diagrams that are given in your NCERT. This is single stranded DNA. Excuse, if you have your NCERT, just open it. Here itself you make on C1, C2, here itself you make on beta C1, because here nitrogen base is binded. If it is adenine, what nitrogen is binded to first carbon? If it is adenine nitrogen base is binded to sugar by glycosidic bond. Which nitrogen is binded to first carbon? Adenine is purine, no? So N9 of adenine is binded to C1 of sugar, right? So here C1, C2, C3, C4 and C5. Fine. First carbon to the nitrogen base, 5th carbon to phosphate group. Third carbon of one nucleotide is binded to 5th carbon of the succeeding nucleotide. Here already one phosphoester bond is present. One more ester bond is required to connect C3 and phosphate group. So here to connect two nucleotides, it is from 5 prime to 3 prime, right? 5 prime, 3 prime carbon groups are connected by phosphodiester bond. Phosphodiester bond. Is that clear, ma? So to form a single nucleotide, you need only 2 bonds, glycosidic bond and phosphoester bond. To form a single stranded DNA or a polynucleotide chain, you need one more bond, that is what? Phosphodiester bond, that is in between 5 prime to 3 prime. So Shakil Khan, you just tell me what to repeat, whether it is nucleotide or polynucleotide chain. You just tell me what to repeat, that I will repeat. Fine. Now you see double stranded DNA. In a double stranded DNA, along with the phosphodiester bond, okay, there is one more bond is binding between two nitrogen bases internally. This bond is what? Hydrogen bond. Hydrogen bond. Is this fine? Clear? One more point to be highlighted. At every polynucleotide chain, one phosphate group at 5' end will be free and one hydroxyl group at 3' end will be free. That itself is your 3', 5' ends of a double stranded or single stranded DNA. Clear, right? Yeah, take it. If this is clear to you, we will move on to length of DNA in different organisms. Shall we move on to length of DNA in different organisms? Here we go. Organisms and length of DNA. Bacteriophage is having 5386 nucleotides. Here he has clearly mentioned nucleotides, means it is a single stranded DNA. If it is base pairs, two bases are pairing, that is double stranded DNA. So here it is what na? Single stranded DNA. Bacteriophage lambda is having 48,502 base pairs, means it is a double stranded DNA. Escherichia coli prokaryotic cell, these two are what? Viruses. This is a bacteria. Haploid content of DNA, eukaryotic cell. In E. coli it is 10 to the power of 6 base pairs. In haploid human DNA it is 3.3 into 9 to the power 10 to the power of 9 DNA, base pairs of DNA. Clear? This is very important. Fix it in your mind, this table. We may expect in match the following type of questions. And you have to be clear on the note, even when numerical type of questions are asked. You have to be clear on the note of, if he is mentioning nucleotide means it is a single stranded DNA. Base pairs mentioned means it is double stranded DNA. Tell me how many bonds are involved to form a single stranded DNA here. How many bonds are involved? Just now we have learned, right? N-glycosidic bond, ester bond, phosphodiester bond. There is no role of hydrogen bonds here. For a formation of double stranded DNA, we have 4 bonds. What are they? N-glycosidic bond, phosphoester bond, phosphodiester bond, hydrogen bond. Just take down for single stranded and double stranded DNA. Okay. Now, so now we will learn about nitrogen bases. As I have told you, we have purines and pyrimidines, right? Total we have 3 pyrimidines beta. I am talking only about nitrogen bases, not about those nitrogen bases which are in DNA. I am speaking about nitrogen bases on whole. Here these are purines. Pyrimidines are homocyclic, they are single ringed structures. Here you have for DNA and RNA different types of pyrimidines. RNA consists of uracil and cytosine. DNA consists of cytosine and thymine. Whereas purines are common both for DNA and RNA, we have adenine and guanine. Look at the structures carefully and take down what are those present in DNA and RNA. Now we will learn the IUPAC names of each nitrogen base, that is also important. Too fast. Here we go with IUPAC names. Important, Abdul. Important for mol bio, important for biomolecules as well. Fine, right? Because in both the chapters we are having this. Fine, right? So here you see 1. Adenine, whose common name is, adenine only, whose IUPAC name is 6-aminopurine. This is present both in the DNA and RNA. Fine, right? Guanine, 2-amino-6-oxypurine. If you want, you can get the reference of the structures also. Adenine, 6-aminopurine, right? Adenine, you see, it is 6-aminopurine. Guanine, you see, 2-amino-6-oxypurine. 2-amino-6-oxypurine. See, here it is 2, at second carbon, amino, 6-oxypurine. Now, pyrimidine synonyms we will see. These are also present both in DNA and RNA. Cytosine, 4-amino-2-oxypyrimidine. Here you see cytosine, 4-amino-6-oxypyrimidine. Fine, right? And then thymine, 5-methyl-2,4-dioxypyrimidine. Okay, here we go 5-methyl-2,4-dioxypyrimidine. Can you see here 5-methyl-2,4-dioxypyrimidine? Fine, right? Then we have uracil, which is 2,4-dioxypyrimidine. Can you see here 2,4-dioxypyrimidine? That's it. Remember, all the structure is not important, but IUPAC names is also important. You can remember this in reference with the structures. Fine, right? Take down, quick. Now, we'll see. Did you finish that? Yeah. Just a sec. Yeah. Any doubts still here? Yeah. Here we go with purine and pyrimidine. Differences between purines and pyrimidines. Simple only, till now what we have discussed, that only will be there. Just go through this, what all we have discussed, that only will be there. Have a comparative analysis. If you are having any doubt, ask me. Need not to take down. Done, ma? Here you take down glycosidic bonds, that is nucleosides, both for DNA and RNA. Those namings are important. You will get in the objective formats. Okay, nucleoside terminology is important, you see. Nucleosides. See here, if adenine nitrogen base along with sugar, this is an RNA. Addressed as what? Adenosine. Guanine plus ribose sugar. Guanosine. Cytosine plus ribose sugar. Cytidine. So, yeah, adenosine, guanosine, cytidine. One is skipping up, that is uridine. That is uracil plus ribose sugar. This is addressed as uridine. Fine, right, ma? Now in DNA, if you see all the nucleosides, deoxyribose sugar along with thymine is deoxythymidine. Along with adenosine, it is deoxyadenosine, deoxyguanosine, deoxycytidine. Take down. Terminology you will forget again, that is why I am asking you to take down. At least you write for one and you just keep hyphens for the other. Cytidine, uridine, thymidine will be little confusing. Note down. Done, ma? Here we go with this phosphodiester bonds. If you see, this is in between two nucleotides, as I already told you how a polynucleotide chain is formed between 3' carbon of one nucleotide to 5' of the preceding nucleotide. Based on the polynucleotide chain, first thing it is Rosalind, Wilkin and Franklin's. It is the two scientists Wilkin and Franklin has proposed X-ray diffraction studies on the polynucleotide chain. Okay. Based on those X-ray diffraction studies, it is Watson and Crick who proposed double helical model of DNA and salient features of the DNA. Clear? So here you see the DNA which is double helical model. First I told you it is in 1869. Frederick Mister identified it as nuclein. Rosalind Franklin and Maurice Wilkins has given out X-ray diffraction studies. These X-ray diffraction studies are analyzed by Watson and Crick. They have proposed double helical model of DNA from which salient features of DNA are proposed and Chargaff's rules were also proposed. Fine, right? Erwin Chargaff pointed out that in a particular DNA, total number of purines will be equal to total number of pyrimidines. Fine, right? Number of purines will be equal to number of pyrimidines. The number of adenine thymine bonds will differ, okay, and number of guanine cytosine also will differ. So the base ratio A plus T by G plus C is constant in all the species. Remember that A plus T by G plus C is constant in all the species. Now we will move on to the salient features of DNA. As here this has come, there is one more ratio. Anyhow in Chargaff's rule we will learn again, but here you remember A plus G by T plus C. The ratio of this will be variable in each organism, okay. That will be less than 1 in prokaryotes, more than 1 in eukaryotes. Salient features of DNA, it is made up of two polynucleotide chains where backbone is constituted by sugar and phosphate group. The backbone of DNA is constituted by what? Sugar and phosphate group. Two chains are coiled in what fashion? Right-handed fashion, and mostly biological form of DNA will be B-form DNA only. We have learned in biomolecules, right? And then the pitch of the helix is just 3.4 nanometers and there are roughly 10 base pairs in each turn. Consequently, the distance between two base pairs is approximately 0.34 nanometers. The plane of one base pair stacks over the other in the double helix. This, in addition to hydrogen bonds, confers to the stability of this double helical model. Clear? These are the salient features. And two more important points. One is complementarity, second is anti-parallel to each other. One polynucleotide chain runs from 5' to 3' and other runs in the anti-parallel direction from 5 to 3. The bases in two strands are paired through hydrogen bonds. These are paired through hydrogen bonds forming base pairs. Okay, a purine always comes opposite to pyrimidine. This generates approximately uniform distance between two strands of the helix. Central dogma we have already listed, followed by DNA packaging. DNA packaging we will look in the next class. Okay. So till here DNA, how is it discovered first, what is the central dogma of DNA, what is the structure of a nucleotide, how a polynucleotide chain is formed, what are different types of base pairs in different organisms, followed by the bonds of both DNA, RNA, right, analyzing X-ray diffraction, salient features of DNA, okay, from double helical model given by Watson and Crick. Till here it is done. In our next class we will be looking into DNA packaging, RNA world, followed by DNA replication. Clear till here? Yeah, that's all for today's class, beta. Thank you.